Well, thank you, Matt, for the very nice introduction, and thank you, the organizing committee, to give me this opportunity to present some of the work we're doing here at our stage, and also to all participants, that are attending this lecture, that attended the last lecture, that are attending, the multiple days of Iowa Swine Day. It's nice that, it's unfortunate that we cannot all be together, but it's nice how this online dynamics work and allows people from all over the globe to attend these talks. So I'll continue talking about disease, but hopefully never going to have to talk about dealing with African swine fever here in the US. So we're going to talk a little bit about PRRS and a lot of what we have done on the host genetic side of it. So I'll start out by saying that it's very important for us geneticists, especially working with diseases, to collect data at a commercial level. Okay, but what does that mean? So if you think of just a typical breeding scheme in swine production, it has this shape, it has this pyramid scheme that has three tiers: the nucleus, it is the top of the pyramid. We have the multiplier and the commercial level. Okay? So from a genetic standpoint, what happens is that here in the nucleus, you think about the landrace nucleus. Okay? And that's where those are our best animals. That's our breeding stock. These animals have data collected on everything of them. The number of teats, feed intake, growth, back fat, loin muscle area, number born alive, and so on. Okay? And that's where genetic selection occurs. The best animals are selected, are identified, are mated to each other, and then they create this next generation of animals to form the population. And the idea, of course, and it works, is that this next generation is better than the previous. So we have this genetic gain. And we expect this genetic gain to move down this pyramid. So we multiply our animals in order to meet the demands of the consumers. Then animals are crossed, so we create our F1 gilts that are sent to the commercial farms. And these F1 gilts are mated, are inseminated usually, let's say, with the Duroc sire, and we have the three cross hogs. Okay? So we expect that these animals at the commercial level are expressing the best genetics that has been gained from their grandparents in the nucleus. Okay? So at the commercial level, we're going to have expression of traits that we selected for. So if you think about the gilt, okay, and becomes a sow, so the animal has, let's say, high number born alive. If you think of the hog, the three-cross hog, well, this animal grows well, has very high feed efficiency, and so forth. But not only that, we also express traits that we don't select for. Okay, so for instance, the ability of the animal to digest high fiber. Okay, because well, at the commercial level, we use high fiber much more frequently that you can think of the nucleus, and this trait is not selected for. Okay, also response to vaccinations, response to diseases, heat stress, and so forth. So the commercial level, that's where we have our product. We also have the expression of traits that are not selected for, but of course those traits that we select for. Okay? But one of the things that happens in swine production in this pyramid scheme is this part here that we say that the genetic correlation between the nucleus and the commercial level is different than one. What does that mean? So think of growth rate. If you measure growth rate in the nucleus and growth rate at the commercial level, not necessarily the best animals that grow in the nucleus will be the best animals that will grow in the commercial level. The reason for this is because we have a different environment, right? So the nucleus is the best environment for the animal to express their traits, right? And the commercial level, well, we also have exposure to diseases, also have stress, different management, and so forth. So we have what we call genotype by environmental interaction. And the other component to it is that the commercial level, we have crosses. So in the nucleus, we have purebred animals, and the commercial level, we have crosses. So there's also gene by gene interactions going on. So that's why the trait in the nucleus is not necessarily the trait at the commercial level, right? As I just explained, we also have traits at the commercial level that are not expressed in the nucleus. And the unsung hero here is genetic relationships. That's the only way that we can actually perform genetic selection. For you to identify the animals that carry the best genetics, you need to know who are their parents, their uncles, their brothers and sisters, and so forth. Okay, that's by using pedigree information. Or nowadays, we can also have the opportunity to use genotypes. And here in the nucleus, animals have pedigree, perfect, well, somewhat perfect pedigree, and most of them nowadays already have genotypes. But as you move down to the pyramid, you pretty much lose all of this information. Very hardly you can get any type of pedigree information at the commercial level. So what does that mean? Is that the commercial level, which is the level that we want to improve for, want to improve our traits for, we actually cannot use that data because we don't have pedigree information. However, with the development of genotype platforms and has been getting cheaper and cheaper, we may now use this information back and identify which are the best, genetically best animals. And just to complicate things a bit more, you know, we are talking about actually somewhat three pyramids because we usually use two dam lines and one sire line. Okay? So the bad of what we have nowadays, as I said, we have limited information at the commercial level. And to perform genetic selection, we actually need individual records. Working with a pen measurement or just a farm measurement doesn't tell us anything because we actually need to know what is what each animal is producing and what is the genetic makeup of that individual. But so to collect individual records, it's somewhat labor expensive. Many of the traits are available. Collecting individual feeding take at the commercial level is very, very rare. Okay? Birth weight or the information can be very inaccurate, like the number of piglets weaned. Right? There is a lot of cross fostering, and you know a foster mother that is able to wean 10 piglets, maybe only one of those actually was born from her. So we need to keep track of this genetic information if we want to perform genetic selection based on that data. Yeah. From the point of view of ability of improving traits, we know that there are many traits that are lowly heritable. And for instance, reproductive traits are known to be lowly heritable. So only 10% of what you see is due to genetics. And I'll talk a bit about this again later on. And of course, many farms have very different ways of manage the farms and the animals and so forth. So all of those things actually impact our genetic evaluations, including effects that we cannot observe, the weather interventions, the animal interactions, and so forth. So all of this happens, and we cannot create our genetic evaluations in a way to capture all of this. But the good, well, as I mentioned, nowadays we have genotyping technologies. We have been using this. The swine industry started using this about 8 years ago. Okay, and with this, we can actually, as I said, we can collect data at the commercial level. Just take a few pieces of fur, samples of fur or of hair from these animals, and we can extract DNA and genotype them for thousands of genes. We are creating new technologies to what we call big data and phenomics. Okay. Nowadays, we are starting to go beyond growth, number born alive, and so forth, and start looking more in physiological responses. We can look at the microbiome, the proteome, transcriptome, and start using sensory data. Luckily, we have been very effective in developing models, statistical models, to deal with these type of data. Many of these data are normally distributed. They are very correlated and so forth, but we have several methods that deal with these efficiently. And of course, with that much data, the complexity of the models, now we need to have computational power, and we sure have these. Okay? So what can we do with this? So the questions are, how can we improve lowly heritable traits? So for instance, as I said, number born alive has a heritability, and heritability is expressed by this little h squared of about 0.10. So think of 10% of what you see in the population is explained by genetics. So all of the other 90% are non-genetic factors. We call them environmental factors. Okay? So one strategy is, well, let's say that we can find another trait that, instead of explaining 10%, maybe explain 60%. Okay? Of genetics explains 60% of that. Okay? And the rest is explained by the environment. But the trait is highly genetically correlated to our trait of interest. So if we change our indicator traits, we are also changing the trait that we actually want. And this is what we call an indicator trait. It's something that indicates something that we actually want to improve. Another strategy is to find, let's say, intermediate phenotype, intermediate trait, or something that you can build on top of the trait of interest. So we have this novel trait here that can explain parts that the genetics cannot explain. And now most of the phenotype is being explained by these traits. So as novel traits, we can use all of those omic big data types that I talked before. Right, this is what we call shotgun approach. Let's try to find something. Or we can also try to work with things that are more related physiologically with the traits of interest. So in case of animal born alive, I think, well, something related to reproduction, maybe a hormone. And in case of disease like PRRS, perhaps the immune response of the animal. Okay? And what's the importance to select for, let's say, disease resilience, just a very general term? I'm glad that this is an audience that I don't have to convince you to do this. And you all remember well, right, since 2013 when we had PED here in the US, and about 10 years ago when we have H1N1, so we had these major costs because of production. You have them also because of zoonotic threats. We have issues with animal health, welfare, and so forth. Diseases are not expressed in the nucleus, right? Or theoretically, they're not expressed in the nucleus. They're expressed at the commercial level. Okay, so we need to get that data. There's a complex definition. Okay, so right, the response of the animal to a given disease may depend how much of the virus the animal receives, how much the virus is being shed, and so forth. So it's very complex to say, hey, this animal is sick. Sickness can have a very huge range of response. But as I'm going to show to you, we have been seeing a lot of genetic variability from the animal in response to its infection. And finally, we need to feed about 29 billion people by 2050. So that's one way that we can think we can help to achieve. So talking about PRRS, if we have a healthy animal, we expect that animal to say, let's say, has a total born alive of 12, one was born mummified, another one was born, well, was stillborn, the other one was born mummified, and out of these 10, this sow is able to wean 9. But in the presence of PRRS, now maybe we still have the same total number born, but now we have 2 stillbirth piglets, then we have 3 mummified piglets, so now we're down to 7 that could be potentially weaned, but they're weaker. These piglets are weaker, and actually you only wean 5. Okay. We know PRRS has been around for a long time. Costs us about $1 billion per year, okay, total. But from the disease itself, in production, we can think of about almost $700 million per year. And with what we have been doing in the previous years is focusing a lot on biosecurity and vaccination. Okay? Biosecurity, we're humans and sometimes we make mistakes, and there's also the luck component, right? Or the bad luck component. And in vaccination, we have not yet been able to create a vaccine that works for everything. Of course, this is a complex virus that mutates all the time, has very, very diverse ways of transmission. Okay, So another tool that we could potentially use is to select animals that are genetically more resilient to PRRS. And what we need to look at is this again this measurement here of heritability. So how much of what we see that the animal produces is due to the host genetics of that individual. Okay. So over the years, Craig Lewis in 2009 was the first one to provide heritability estimates for PRRS response, and since they did that, started to have lots of publications, okay, especially in the previous 5, 7 years, right? And most of this work here is actually done at Iowa Swine. So some people have looked at growth and pathogen burden, very different range of depending on the trait you're looking at, of heritability. Reproduction, as I said, is usually lowly heritable, and also immune response. So I'll try to go over some of these studies today. So start out talking about the first study that we did on reproduction and immune response, which I'll call PRRS Outbreak Herd study, PRRS Outbreak Herd number 1. And as you can see here, so this is a traditional performance in a cell farm. So we have the number born alive in blue, number of alive 24 hours in red, number of stillborn and mummified piglets in green and purple respectively. And then sometime you have a huge decrease in number born alive and an increase in number born dead piglets, right? Very typical of a PRRS outbreak. So the outbreak was detected. Blood samples were collected on all of these animals. There were about 600 sows in the farm. And these animals were collected, the blood samples were collected to do a PRRS ELISA test and also genotyping on these animals. Okay? But as I mentioned, we are always interested in measuring how heritable these traits are. Okay? So how much of genetics explain the variation of these traits? So heritability for reproductive performance during the PRRS outbreak, slowly heritable, between 7 and 9%. Right. Remember, so to be lowly heritable of all the variation that you see, just a somewhat small component of it is due to genetics. Nonetheless, we can still select and improve performance under this environment. But we use the PRRS ELISA data and we measured what we call sample to positive ratio, which from now on I'm going to say ASP ratio. And we saw that the heritability for aspiration was quite high, of 45%. So about half of all the variation that you see is explained by genetics. And so we have never done that. No one had ever done that. And we had no expectation as far as the value that we could obtain. So we naturally asked ourselves, so what would be the relationship between reproductive performance under a PRRS infection and antibody response? Okay, so reproductive performance under PRRS infection is lowly heritable. ASP ratio, so antibodies to PRRS, is highly heritable. And that's what we got. So we looked at the genetic relation between these two traits. And we observed that for number born alive, it was quite high and positive, and for number of stillborn piglets, it was quite high and negative. What does that mean? It means that animals that are genetically superior to produce more antibodies, like sows that have high antibody response to the genetic level, they should also have greater number alive during the outbreak, and also therefore lower number of stillborn piglets during the outbreak. Okay? In order for you to visualize a bit how this translates into selection, just think that over the generations we are selecting for a number born alive. Okay, and that's okay, over the years, the overall performance is increasing. Okay? But the idea is, well, if antibody response is highly heritable and highly genetically correlated with number born alive, I can also change number born alive just by changing ASP ratio. So if we change ASP ratio, this is how much of number born alive they were increasing. Okay? So it's more efficient to increase number born alive by looking at antibody response to PRRS compared to directly selecting for normal born alive. And ever since, we started to suggest the SP ratio, so antibody response to PRRS, could be an indicator trait for reproductive performance during the outbreak. So we observed that these two are highly genetic correlated, but these animals also had genotypes for. So I believe that out of all participants, at least one of you have done 23andMe, right? So looked at your whole genome. And that's what we do genotypes to. We do 23andMe with every animal, okay? But pigs don't have the 23 pairs of chromosomes. So we genotype these animals for thousands of genetic markers across the genome. And we can do what we call genomic scan. So we can try to find across the whole genome, right, across all the chromosomes, if there are any regions that contribute to variation to antibody response, to SP ratio. And the higher is this dot, a greater effect of that region of the genome has on SP ratio. So we found here two regions on chromosome 7. One explains 16% of the genetic variation of SP ratio, another one quarter of it. Okay? So in total, about 40% of the genetic variability in aspiration in these animals could be explained by two regions. The interesting part for this region here is that it's located in the MHC, which is the major histocompatibility complex region, which is the region in mammals that control pretty much all of the immune system. Okay? So we found that we're quite excited. We had no idea that antibody response to PRRS was heritable, no idea that it was highly correlated with number born alive, and have no idea what were the genes, the parts in the genome controlling this trait. That study was actually part of this bigger study called the Gilt Declination Project, which we had about 3,000 crossbred replacement gilts from 7 breeding companies sent to 23 different farms. Okay, so very large project and lots of genetic and environmental diversity. Okay? And we measured SP ratio on these gilts, okay, to PRRS SP ratio, and this is the result that we got. Okay, so they are very similar plots. Okay, the one at the top is the one that I showed you before, was just one purebred population from one breeding company. Then we have this, a large data set with crossbred animals with different genetic makeup from different genetic backgrounds too. But the two regions popped up in the analyses for the same trait. So we believe that certainly the MHC controls a good portion of response to SP ratio. Okay, so we're developing more of our knowledge of what controls SP ratio, what is the genomic basis of it. So we know that reproductive performance under PRRS is lowly heritable, is highly genetically correlated with antibodies PRRS that is highly heritable, and it has two major genomic regions controlling it. Okay? But these genetic markers, let's say that the most important part of using genetic markers is that we can actually do selection based on these. So can we predict, the response of SP ratio based on the genomic information that we have with these animals? I don't want you to look at all of the bars, okay? Only look at the colors for only the Iowa State colors, which are the best ones and which are the methods developed here at Iowa State compared to the white one. Okay, so what I'm showing to you are the accuracies of how good we can use these genetic markers. And each group of bars just represents a different set of the number of genetic markers. But I want you to look at this here, this fourth group of bars, which we're using 269 genetic markers on those two genetic regions that I talked about. And we have an accuracy of about 50%. Okay. So by only using a few genetic markers, we have a very nice accuracy of identifying which are the best animals So therefore, we can potentially use genetic markers to identify which are the animals that have the highest antibody response to PRRS. And of course, we are we are having the hypothesis and moving forward thinking that having high antibody response is good because that's also highly genetically correlated with the performance. Okay, so we want to increase genetically the antibody response to animals and expect that this increase in antibody response results in increased performance during disease. Okay? So we constructed a bit more in our diagram, and we found that using genetic markers, we can get some nice accuracies to predict antibody response using genomic data. Okay. So from these two studies, we got these two overall understanding of the basis of SP ratio. First, from our outbreak study, okay, so during natural infection, SP ratio is highly heritable. During natural infection, it has high correlation with number born alive during infection, and also is controlled by two major genomic regions. From our other study, I didn't show this result, but heritability was higher, 0.47, again controlled by two genomic regions, and we could see that we could actually use genetic markers to predict SP ratio. But one interesting part of this guilt acclimation study is that we had no idea of what happened during acclimation. That's why I have these question marks here. We don't know if animals were actually infected with the virus or if they were only vaccinated. And when we looked at the data, out of the 23 participant farms, 20 of those mentioned that they used PRRS vaccination. So we created a hypothesis that, well, can we get these same results just by vaccinating animals? Okay. That's a project that I'm going to show today that my PhD student Leticia Sengler has been working on. So we had a trial performed at two farms in North Carolina with about 900 F1 gilts. All of them were vaccinated with a commercial MLV vaccine. And 52 days later, they had blood samples collected. And with these blood samples, we measured SP ratio, and we genotyped them for 50,000 genetic markers. So this is the data. This is the SP ratio data. This bar here represents what many people use as the cutoff to identify positive animals. So out of the 900-ish animals, we had only 12 that had values below 0.4. But I don't like to say that they are negative. I like to say that they are low responders, okay? Because all of them were vaccinated, so all of them are positive. The fact that they have low SP ratio is the fact that, well, they're genetically low responders. And we got moderate to high heritability of 38%, a little bit lower than the other two, but still very substantial. Okay, we performed the same genomic scan, and this one is a bit cleaner, but I'm sure that as you can see here, there is a dot there. And this is, again, the major histocompatibility complex region, the MHC. But this time explain actually 30% of the genetic variants. But the interesting part is that, remember that I told you that there were two genomic regions, and for some reason, that other region never popped up in this population. And we have done additional analysis, and we believe that that region, the genome for this population, actually doesn't segregate. There is no variation there, actually, that could be picked up. So here, just to compare, right? So this is the region that I was talking about. The second region here did not show up. But the major histocompatibility complex showed up and explained 30% of it. Okay. So we did the same drill, right, from our other studies, and we try to see, hey, can we predict genetically who are the best animals? So this is what I'm plotting now. Here is using all of the 50,000 genetic markers. You're only using about 200 genetic markers in the MHC, not even, sorry, 100. And here's using the rest of the genome. And just for comparison, with the results that I showed you before. Actually, in the previous results, we got about 0.45 using the hall markers. In this study, we got about 0.7 using all markers. Before, we got about 0.48 using only the MHC. This one was about the same. And before, we got only 0.10 accuracy using the rest of the genome. This one we got greater. And this is very easily understandable because in the previous study, we're actually using data from 7 different breeding companies. And in this study, they all came from one single breeding company. So we're actually making use of animals that are much more genetically related to each other. So if they're more genetically related to each other, we can find genetic effects much more easily. Okay? And that's actually what is done in practice, right? A breeding company uses their own genetics to identify the best animals and so forth. So this is actually a much more realistic result than the previous one. Okay, but the whole reason why we're looking at aspirate ratio is because we believe that this has some relationship with reproductive performance. So we looked at the relationship between aspirate ratio and number born alive first parity. But these animals at first parity, they were all healthy in a sense that there was no first outbreak going on, okay? Which is not the situation that we had before. Okay? The first study that I showed you, we're looking at relationship between aspirate ratio and number born alive during an outbreak. Now we're looking at the relationship between PRRS MLV aspirate ratio and reproductive performance without PRRS. But nonetheless, we also get this very nice high correlation of 0.61. Okay, and just to compare, before we got 0.72. So we started thinking that, well, perhaps this relationship holds regardless of the disease, which would make this tool far more applicable. Okay, and then in the meantime, while we were having that trial, there was an outbreak with one of our collaborators, and they shared some of the data with us. We kind of regenerated the same idea. Okay, and all of this, and this is just a summary. Okay, so this is the study done by my former master's student, Philippe Hickman. So we worked with Duroc and Landrace populations, and what I want to focus here are 2 components. One is the heritability of aspirate ratio was slightly lower than we had before, and we start to think that the date of collection has something to do with it. But we got, again, this very high genetic correlation with number born alive. So we validated our previous results that these 2 traits are related to each other. So putting all of these together, before I move to the final topic, okay, so we can think of exposure of these animals to PRRS through 2 ways. One is through an outbreak, right, an uncontrolled natural way that no one wants, and through PRRS vaccination. Okay, so that's usually most of the farms use vaccination for PRRS and GIPS. Okay? So when we look at number born alive and antibody response to PRRS, number born alive is lowly heritable. We know that. And heritability for aspirate ratio, it should be somewhere between 0.32 and 0.45. Okay. When we look at PRRS vaccination, from the study that we got on vaccination, we got 0.38, a heritability for aspirate ratio, and again, number born alive, low, well, less than 10%. But the most important part of it is this relationship. The genetic correlation between antibody response and number born alive is quite high, something between 0.65 to 0.72 during the outbreak, and the estimate that we got in healthy animals, right? Remember that this number born alive is at a parity 1. These animals were not under a PRRS outbreak. It's also quite high. So since these two studies actually came from the same breeding company, the vaccination study from Leticia Sengard and this outbreak, the second outbreak study from Philip Hickman, we were able to get a genetic correlation between antibody response to outbreak and antibody response to vaccination. We saw that was quite high. I was expecting to be greater than that, but it's not one. Okay, so what does that mean is that genetically, a response to an outbreak shares most of the same components as a response to vaccination, but they're not exactly the same. They're still different somehow. Of course, it could be because of the virus, but it could also be because of the genetic makeup of the animals. Both studies that we have on outbreak were done in purebred animals, and in vaccination we have F1 gilts. So we also have a cross in the purebred situation. We also looked at the relationships between aspirate ratio due to outbreak and performance in commercial animals. We couldn't find anything, and also the performance during an outbreak and aspirate ratio due to vaccination, and also we couldn't find much. Okay, so we're still trying to better understand what will be the most efficient way of use this selection tool. Okay, so this is one of the potential ways of novel ways of improving performance of PRRS exposed animals. And the other way that I want to briefly talk about is that in the same PRRS vaccination study, okay, so this is the design that I showed you before. The animals were vaccinated, and 52 days later, they had blood samples collected. We also had animals having vaginal swabs collected at day 52 and day 4. Okay, so animals were vaccinated with PRRS, and a subset of them, 302, had vaginal swabs collected 4 days after the vaccination, because we're trying to mimic some time after if there is an outbreak going on. And then 52 days later, just because there was the day that we also collected blood samples to measure antibody response. So we collected vaginal swab and we looked at the microbiome of these individuals. Here, the goal of this figure is not for you to read it, just for you to have an overall idea. Okay? Each of these bars represents one bacteria in these animals, okay? And the different colors represent the day of collection. And here at the x-axis, we have heritability. So just want to show you that we have some bacteria that have low heritability, like this, like this, like this, and others that have very high heritability, like this one, like this one, like this one, like this one. Okay, what does that mean? It means that the vaginal microbiome of pigs is partially controlled by the host genetics, so the animal genetic capacity of increasing the abundance of these bacteria. But some are not controlled by the host genetics at all, just the environment. And the other piece of information is that, well, it really depends the time of collection. Okay? But the animal isn't evolving, the animal is acclimated to the environment. There are other things playing a role that we'll never be able to answer using these type of data. But what I want to show to you is that we can use these bacteria to try to find relationships with performance. So when we looked at the relationship between the abundance, how much bacteria animals have in aspirate ratio, we can find that animals that have very low antibody response, those that I mentioned to you before, those low responders, we have these animals, they have high abundance of some of these bacteria here. So we can think that, well, perhaps these are bad bacteria. And those with high antibody response, so they are genetically very high up to have antibody response, they also show greater abundance of this one. Okay, but that's one approach. That's actually an approach that I don't like much of using, try to find what is the one bacteria that is associated with that. But we can use, remember when I said at the beginning of the talk about those complex models and correlations and so forth, we can actually use the whole data and try to classify individuals, okay? And we can perfectly use the bacteria data, the microbiome, the vaginal microbiome data, and classify animals in being high antibody responders based on data collected day 52 or based on day 4 from those with low response. See, we create them, we classify them in their groups perfectly. And we also did the same approach using reproductive data. Okay? So can we see can we see difference in the microbiome of animals that have very low, let's say, total number born compared to very high total number born? Do their bacteria differ? And this is the overall results. And again, I'm going to use the same explanation that this is one approach of doing one at a time and trying to find what are the bacteria that differ between them. And I don't think that that's the most efficient. I think it's much better to use the whole data together and see, hey, if we have this group of bacteria, can we tell if this animal will be high or low performer? And when we did this, we actually got a perfect classification for all the traits. We were perfectly able to classify the animal as being high or low performance for a number born alive, how low performance for total born, how low performance for a number born dead, and so forth. Well, perfect classification for all, but these ones here. So number born alive, when we use only data from the first day of collection, reminders from the first day of collection, or number win from the second day of collection. But classification was not perfect, but the accuracies were not the lowest one was 89%. May not be perfect, but 89% is damn good. And the other point that I want to highlight is this, that moving away from that approach of trying to find one single bacteria at a time, we can see that actually one bacteria never worked. I actually used between 14 and 16 bacteria to efficiently classify these animals, showing that the microbiome is not one thing, it's not one bacteria that could be associated with response, but actually the group of them, because they work together in complex ways, and we could use that complexity together in order to identify which guilds would be the best cells. Okay, so remember that these animals had vaginal microbiota collected before they were cells, before they were vaccinated. So they can actually be screened for you to identify which will be the best animals. So you can cope the bad animals, or you can actually make the bad animals perhaps being that good as the other ones. Okay. So with the use of antibody response to PRRS and that we have done a lot with it already, and we have seen many of interesting relationships, and the use of the vaginal microbiota that we still have a lot to learn, we expect that even the presence of PRRS, okay, we transform this very sick animal that produces very badly in a very strong cell, and that even with PRRS, this animal is so robust, so resilient, that her performance will be as good as the animal without the disease. Okay. So just to wrap it up, as I showed you, reproductive performance is quite complex, not only from the genetic point of view, right? It's slowly heritable, but why is slowly heritable? It does not depend only on the animal to get pregnant, right? The animal needs to cycle, needs to be inseminated, and so forth. So it's quite complex trait, but it's very important for the industry. Very profitable part of it is the number born alive. Okay? So we need to improve that. We're showing that we can actually data collected at the commercial level can be used with success. So the SP ratio should not be observed at the nucleus, okay? But it is constantly observed at the commercial level. The microbiome data was collected at the commercial level, so there is a great potential there. As genomics gets cheaper and cheaper, soon, okay, this will be a very standard procedure to genotype all animals at the commercial level. So this will accelerate genetic gains much more. And we're looking at these two novel traits. Okay, so antibody response to PRRS measured as sample to positive ratio has moderate to high heritability. We got high genetic correlations with reproductive performance. So during the outbreak, This correlation is between 0.65 and 0.72. But even without an outbreak, we also get a very high due to vaccination, being 0.61. The most genomic important region is the major histocompatibility complex, which controls the immune system of mammals. And we also showed you that genomic prediction works really well. You can actually use genetic markers and predict with high accuracy which are the animals that will have better immune response. The microbiome, remember, we're not looking at one trait, we're actually looking at a couple of thousands of bacteria. So there is a huge range of heritabilities. I only showed you actually 100, the first 100, but there is a huge range from low to highly heritable. But it was very good to predict, well, perfectly to predict the groups of antibody response. But more importantly, is that we could collect data on gilts before they were even inseminated, okay, and predict with very high accuracy, if not with perfection, which would be their fairing performance group. Okay? But so with that, I'd like to thank the members in my lab, especially Leticia and my former student, Philip Hickman, that presented some of the work here, all the Iowa State collaborators that helped me with a lot of this, Dr. Jacques Deckers, Dr. Rohan Fernando, Dr. Stefan Schmitz-Esser, and Dr. Daniel Linhares, all of the collaborators from other institutions, and my industry partners, and of course, the industry partnership with Smithfield Premium Genetics and PigGen Canada, which are a group of genetic companies that are also located in Canada, and of course, the huge support of the funding agencies. And then with that, I'll be glad to entertain any questions. Thank you.